Assalamu alaikum dear students. Today I will start a new chapter, Cell and Tissues of class 9. The topic is microscopy. First I will explain what is microscopy. It is the use of microscope. We use microscope to observe the objects which we cannot see through a naked eye. So we may define it as the technical field of using microscopes to view samples and objects that cannot be seen with the unaided eye. The terms related to microscopy are, the first one is resolution. The ability of microscope to separate or distinguish small or closely adjacent images. Or we can say that it is the clarity of the image of the specimen. Now look at this picture. The first one is not resolved. So in this one you can see a single dot. While the one which is just resolved, you can see two dots which are attached to each other, while the third one which is fully resolved, the two dots are at a distance from each other. The second term is magnification. It is the process of enlarging the apparent size, not physical size, of something. So the specimen which is placed in the microscope, the actual size of that is smaller, but the microscope gives us a bigger image of that specimen. History of light microscope. The first compound microscope was developed by Zacharias Janssen, and he was from Holland. He introduced this microscope in 1595. He used two lenses in this microscope which were placed in a tube-like structure. At the two ends of the tube-like structure the lenses were fixed, and the magnification power of his microscope was from 3x to 9x. Light microscope was also developed by another biologist, Anthony von Leeuwenhoek. He used one lens in his microscope and he developed it in 1670. This is the picture of his microscope. The magnification power of his microscope was 200x and the resolution was twice of the best compound microscope. And his microscope was better for the reason that he used better lenses than others. The light microscope. You are familiar with this type of microscope because we use such type of microscope in our school and colleges laboratories. In this type of microscope, the specimen is placed at the stage and the source of illumination is light. The light passes through the specimen, due to which the image becomes clear. The nature of lenses is glass. Glass lenses are used in this type of microscope, one in the eyepiece and other in the objectives, and there are different power lenses in the objectives. According to requirement, we can use different type of glass lenses. The one magnify the image of the specimen, which is further enlarged by the other lens. The magnification power is 1500x and the resolution power of this microscope is 0.2 micron meter. Electron microscope. The source of illumination in the electron microscope is beam of electrons, and the nature of lens in this type is electromagnetic lens. It magnifies the image of the specimen up to 250,000 times, while the resolution power is 0.2 nanometer. There are two types of electron microscope. The first one is transmission electron microscope. Beam of electron is transmitted through a specimen in this type of microscope. Its resolution is better than light microscope, and this type of microscope is used to see the internal structural details of the specimen. The second type is the scanning electron microscope, which produces images of a sample by scanning the surface with a focused beam of electrons, and it is used to see outer surface details of the specimen. Now, differences between the light and electron microscope. The first one is that source of illumination in light microscope is light, while in electron microscope it is beam of electrons. The nature of glass lens is glass in light microscope means that the lens is made up of glass in light microscope, while in electron microscope the lens is electromagnetic. The magnification power of light microscope is 1500x and that of electron microscope is 250,000x. The resolution power of light microscope is 0.2 micrometer while that of electron microscope is 0.2 nanometer. Another difference is that in light microscope we can observe the life activities, while in electron microscope we cannot observe the life activities because in this the specimen is placed in vacuum chamber, and in vacuum life is not possible. Survival of a living thing is not possible in vacuum. That is why the life activities cannot be observed in the electron microscope. Thank you, students. This was for today. Allah Hafiz.